Hi, everyone. So, whenever we're performing research and we're trying to analyze our data, one thing we want to be very certain to do is to form our questions and clearly communicate our research interests to other people. Oftentimes, we get bogged down in the statistics and we kind of lose track of what questions we're actually answering. So, in order to facilitate better question asking, I oftentimes like to do something called a physical layout and a statistical design. So, what we do is basically draw out our experiment and then translate that drawing into a statistical design that allows us to clearly ask questions. So, let's start with a question. Say I want to ask, all right, I need to get my pen working. Let me switch pens really quickly. This one doesn't seem to be working quite as well. There we go, much better. So I want to ask the question, does the effect of light levels on SHMU mass depend on the population of the SHMUs? So you should know that SHMUs are made up objects. They're not anything. But oftentimes we get, again, kind of held back by thinking about the types of animals or the types of plants or the types of data we can collect. Well, the shmoos aren't any type, they're just made up. So this kind of just shows you that we can use anything we want to answer questions. So my question is, does the effect of light levels on shmoo mass depend on the population of the shmoos? So this particular question, you've not learned this yet, but you will soon, has two independent variables. Our first one is shmoo population, and our second is light availability. Okay? So within each one of these variables, we're going to have two levels. We're going to have, let's see, we'll stick with West Virginia populations and Kentucky populations. And we're going to have full sun and shade. Now, these are very generic values, full sun and shade. If you were doing an actual experiment, you'd want to quantify those a bit more. But for this example, I think it'll work fine. So, the first thing we need to do is draw out something I call a physical layout. So this just is what it's actually going to look like in the field. All right. So let's say we have our shmoos, and I'm going to make our West Virginia shmoos. I guess I will make them gold because of WVU. We're going to have 5 replicates, 1, 2, 3, 4, 5, in each cell of our design. So we have 10 total West Virginia shmoos. So gold equals West Virginia. And since I'm from Kentucky and we have Kentucky bluegrass, we're going to make our blue color equal Kentucky populations. So I have some blue shmoos. All right, so what's my total number of observations for this this experiment? Well, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So my observations is 20, but I still haven't put my second variable in yet. So what I want to do now is look at light availability. So each one of these groups gets a different amount of light. So our West Virginia shmoos get full sun, and one group of our Kentucky shmoos gets full sun. Then we see that the other group of our West Virginia shmoos gets shade. It gets very little sun. And the same thing with our Kentucky ones. So what we're doing is we're taking two different populations, West Virginia and Kentucky, and we're applying two different types of light availability, full sun and shade. And we want to make sure that each one of our populations receives same or similar treatments, okay? So our replication for this design then is what? Well, our replication is the number of individuals in the smallest cell of the design. So for example, we have 5 Kentucky individuals being affected by the shade, 5 being affected by the full sun, and the same thing with WV. So our replicates are 5. Okay? So that's our physical layout. We can see that, you know, each one of these groups of individuals are being affected by different sunlight. So what about our statistical layout? Well, we can take what we did in our physical design and put it into a statistical diagram that'll make it easier for us to understand. So what we do is we put each one of our independent variables on either axis of this. So we're going to have West Virginia shmoos, so West Virginia and Kentucky. These are the two levels of our variable, which was shmoos. X2, our second independent variable, was light availability. And we had two levels. We had full sun and we had shade, right? Okay. So now you can kind of see what I was talking about replicates. We had 5 West Virginia individuals being subjected to full sun, 5 West Virginia individuals being subjected to shade, and the same with Kentucky, 5 individuals subjected to each light source to give us a replicate number of 5 and a total observation number, which we'll do with a big N, of 20. Okay, so just to go back over this to make sure it's clear, our first independent variable, x1, is shmoo population, and we had 2 levels, West Virginia and Kentucky. Our second independent variable was light availability, and we had 2 levels, full sun and shade. Our dependent variable, we don't draw on this figure. We just know it based on the question we asked. So our dependent variable, which was y, was mass of shmoos. And we'll say in grams. Okay? So by using this design, we're asking the question, excuse me, does the effect of light levels on shmoo mass depend on the population from which the shmoos come? Okay, and that involves 2 independent variables and 1 dependent variable, and that's the mass. So this is just a very simple way to maybe look at your project a little differently. If you're having trouble asking questions about the project or figuring out how many observations you have, how many replicates you have, try to draw it out. Try to draw it out how it looks on your lab bench or how it looks in the field, and then try to translate that drawing into a statistical design. Thank you.